Good afternoon, students. Myself, Dr. Ghanesham Chavan. Today, we will discuss the topic the transfer of or transportation of the liquid across the cell membrane. So, in that, the molecules movement occurs across the cell membranes. So, in that, we will see the passive transport, active transport, endocytosis, and exocytosis. Now, we will start from the passive transports. So, what is passive transports? It is a transport where no energy is required. Means there is involvement of no any glucose molecule, no any ATP will form or no any ATP will required for this. So no energy is required. It occurs without any energy, without any ATP molecule, and move due to the concentration gradient. So here the concentration of the solvent decides its movement. So difference in the concentration, pressure and charge. So here the concentration of that solute occurs into the solvent from higher concentration toward the lower concentration, and also it depends on the pressure and the charge. The move to the equalized gradient means it will produce the concentration gradient or equilibrium condition it will attain. Like if you will see in case of any color solution, when you will start to add some color, so after few minutes that color will be dispersed and that color will be mixed uniformly into your solvent, and uniformly color will be distinctly display on that typical solution. So high moves toward the low. So from the higher concentration gradient, it will move toward the low concentration gradient. So in this fashion, it will produce the equilibrium condition. Now here we will discuss types of passive transports. So there are three types of passive transports are available here. In that, first is diffusion, second is osmosis, and third is facilitated diffusion. So in that, first is diffusion. What is diffusion? Diffusion is nothing but the molecules move to the equalized concentration. So in that I will put some example in which you will come to know how diffusion occurs. So in the diffusion, what you will see? So in one beaker there is a water. The beaker is filled with the water, and in that we will add the lump of sugar. So when you will add that lump of sugar, what will happen? It will go to the bottom of that container, and from that place the particles will start to disperse, they will start to disintegrate, they will start to move to each and every corner of the solution, and after a few minutes they will increase their speed, and due to that, due to this kinetic energy which can be given by these solute molecules, due to that kinetic energy they will increase, energy will increase, and in this fashion it will go to the at each corner of the solution, and after a few minutes the whole solution will be the uniform solution. Means the concentration gradient from the higher concentration it have moved to the lower concentration, and this typical process is called as diffusion. So here in this figure you can see that the whole beaker is filled with the sugared solution. Means that sugar solute has been dispersed or has been uniformly distributed all over the solvent. So this is the example of diffusion. So in this fashion, whatever drug or whatever the salts or whatever the electrolytes, whatever we are ingesting or we are inserting into our body, so uniformly they are distributing, because diffusion is also going on into our body. That's why the volume of distribution is there. So whatever drug we are giving to any person, so it can be uniformly distributed into the body. So that depends on the drug, what is the volume of distribution of that drug. If volume distribution is more, means that drug will reach to your each every or maximum part of your body, maximum volume it will cover of your body. So in this fashion, same thing will happen with your body also. Now osmosis. What is osmosis? Osmosis is nothing but the special form of diffusion. Fluid flows from the lower solute concentration toward the from higher salt concentration toward the low salt concentration, and often involves movement of water into the cell and out of the cell. And in this osmosis, the same process is there. From the higher concentration to the lower concentration, the solute will flow, but it flow through the one specialized semi-permeable membrane. Like in our body, the plasma membrane is nothing but the specialized semi-permeable membrane, and across that the solute or the liquid will be transferred, and that process is called as osmosis. So osmosis is always running in our body, always going on into our body. Now solution differences and cells. So solvent plus solute is called as solution. Now here we will discuss the various types of solutions, and what are their consequences or what are their correlation with our body or with our body solutions. Now first is hypertonic solution. So hypertonic means is nothing but the tone, means concentration, means the solution's concentration is less than our body concentration. What is our body concentration? And we will see afterwards. So hypertonic solution, it is the tone or concentration of the solute that is lower than the body fluids. For example, blood or lymph or plasma. So all are having the they are about same tone. They are having same concentration. So this the solution which are having less concentration, they are called as hypertonic solutions. And the solutes in the cell more than the outside, means here in this solution the less solutes are there, less solutes inside the cell, and outside there is more solutes are available, and due to which outside solvent will flow into the cell. Now next is isotonic solutions or isotonic, means they are having the same tonicity, means same concentration gradient with our body fluids. For example, blood or other body fluids or swab or any other body fluids. So solutes equal inside and outside the cell. So that's why whatever the saline solution or whatever injectables that is going to be instilled or that going to be inside in our body, so that injections are having the isotonic solutions. Means for example, normal saline is having the concentration of 0.8%. So that concentration is isotonic to our body concentration. Or dextrose, 5% dextrose, so it is having near about same concentration as our body is having. So that's why if you will inject that solution in your body, nothing will happen to our RBC or WBC or platelets. Now next is hypertonic solutions. Means these solutions are having more tonicity than our body fluids or blood. So these solutions, so these solutes greater outside the cell, means inside they are less, but they are greater outside the cell, and these solutes are having ability. The fluid will flow outside the cell, means from the cell the fluid will go outside. So in this fashion, there are three types of tonic solutions are available inside the cell. Now we will see how they are working with our RBC here. So as we saw into the hypertonic, the solutes are greater outside the cell and fluid will flow outside the cell. So due to which, so whatever fluid which is inside the cell, it will go outside, it will move outside, and due to which the RBC has created, the shrinkage of RBC has been occurred, means they are contracted, constricted, and the fluid will go outside and into the isotonic conditions or isotonic solutions. The RBC will retain their phase, it will retain their shape, which is round shape. So it is called isotonic solutions or isotonic conditions. And in case of the hypertonic solution, so as we saw in hypertonic, what is there? Solutes in cell more than outsides. So means inside the cell there are more solute and outside solvent will flow into the cell. So due to which what will happen? So outside, whatever outside the solid is there, that will go inside, and due to which the RBC will swell. You can see here the RBC has been swollen. The cells will swell, and if it will increase that solution again, so after a few time, the RBC will be burst. So lysis of cell will be occur, and net water movement inside the cell. So in this fashion, we have discussed the hypertonic, isotonic, and hypotonic solutions. So low solute concentration in extracellular fluid. So in this fashion, the RBC will be tackled by these solutions. And what is the effect of these hypertonic, isotonic, and hypotonic on our RBC or other body cells? Now we will start from the facilitated diffusion. Now that facilitated diffusion is nothing but it is the passive diffusion, it is a type of passive diffusion. It differentially permeable membrane is used here, and the ion channels help molecules or ion enter or leave the cell. And channels usually are transport proteins. Aquaporin facilitates the movement of water and no energy is used. So here, see it occurs into the permeable membrane. For example, our plasma membrane. So in that plasma membrane, the channels are available. The various sodium channels are available. The chlorine channels are available. The potassium channels are available. So across the channel, the transportation of ions exchange will occur, and in between the phospholipids into the semi-permeable membrane of our plasma membrane, there are the aquaporin receptors are available, aquaporin channels are available, and through that aquaporin channels, the movement of water will occur, and due to which the transportation of that water or that solid will occur. So here in this figure, you can understand how it works. The process of facilitated transport. So protein binds with the molecule and shape of the protein changes. So once that specific protein will bind with the molecule, for example, G protein in G protein coupled receptors, specific protein will bind with that molecule or any agent or chemical. So it changes its shapes and molecule moves across the membrane. So in this fashion, so outside the cell, the solid molecules are there. So they are binding here, and due to which they are enter into the cell. So here the transport proteins are available, and this proteins is going to be transport through this channel. So specialized channel mediated transport is there. So due to which the transportation occurs from outside the cell to the inside the cell, and from the high concentration gradient to the lower concentration gradient, the diffusion will occur, and it doesn't require any ATP molecule to be utilized. It doesn't require any energy or any it consume any energy. So in this fashion, in process 2, it will start to enter through the transporter, and here in the figure 3, this molecule has been entered completely inside the cell. So in this fashion, the facilitated passive diffusion occurs into our body. continuously. Now we will discuss the active transport. So here active transports require the ATP molecule. It requires the energy. It requires the hydrolysis of the ATP molecules. So molecule movement will occurs. As I told, it requires the energy against the concentration gradient. And the example is sodium potassium pump. The sodium potassium pump always work in your body. It channelized by the sodium potassium ATPase is the enzyme, and also it is called as sodium potassium ATPase pump. And due to that enzyme, it works on this pump, and where the exchange of these ions will occur. So the potassium pump channels cell membrane, and ATP can get converted into the ADP, and this phosphate has been released, and due to which, so whatever the sodium ions are there which are inside the cell, so that will comes out, and whatever the potassium ions are there which are outside the cell, that can be get entered. So here the exchange of electrolytes will occur, exchange of ions will occur. It can be occur due to the sodium potassium ATPase enzyme, and due to activation of the enzyme, there is consumption of energy ATP. And here in figure 2 you can see the sodium ions is start to move upward, and in this second phase, they are getting outside, and potassium start to get entered inside the channel. So now they are, this is the channel and it is a gate. So gate can be opened due to the enzyme only. When the enzyme will be activated, the sodium potassium pump will be activated at a time only, the gate will open, and when the gate will open, then and then this reaction will occur, this exchange of process will occur. And in the third figure you can see there is complete exchange of these electrolytes has been occur. So the three molecules of sodium has been exchanged with the two molecules of potassium. So in this fashion, so this ions has been opened, as ions has been exchanged sodium with the potassium. So in this fashion, this sodium potassium pump always work on your heart, always work on your blood. So there is continuously exchange occurs. It also works in your kidney. In kidney also this pump is available. In case of potassium sparing diuretics, they are working on this principle, because your body want this potassium. So potassiums are get inside, and these sodiums are throw outside. Now endocytosis. Before endocytosis, again we will discuss the another pump. It is called sodium hydrogen pump, which occurs in kidney. Sodium hydrogen pump, and in that pump also the same exchange will occur. Sodium and hydrogen will be exchanged. So there are sympotors and antipotors. What is sympotors? Sympotors are nothing but the movement of electrolytes or movements of ions occurs in one direction. It is called sympotors, and another is antipotors. When the ions are moving in opposite direction, it is called as antipotors. So that is also one of the example of active transport which occurs in our kidney as well as in our heart. So constantly they are moving, they are doing their work. So in TCT, the sodium hydrogen pump will occur, and at your collecting duct in the nephron in kidney. So at that space you will find this antiporter. So in this fashion, this concentration will be maintained, or concentration gradient from higher concentration to the lower concentration occurs in active transport also. Now endocytosis. What is endocytosis? It is nothing but the process of getting inside the cell to whatever the extra material from outside the cell to get inside. So that process is called as endocytosis. So incorporation of the foreign material from outside to the inside. So movement of that large material, particles, organisms, large molecules. So these all are get engulfed, get inside, and movement is into the cell. Movement occurs inside the cell. Types of endocytosis. There are two types of endocytosis will occur: bulk phase and the receptor mediated specific phase. Now here the process of endocytosis. When the plasma membrane surround materials and the edges of membranes meets, the membrane fuse to form vesicles. So here two types of endocytosis will occur. In that, first is phagocytosis. So in this process, the foreign pathogens or the foreign particles or the bacteria or viruses or the elements which are pathogenic for us, which are dangerous for us. So especially this type of process can be done by the macrophages. Macrophages are doing this phase. What they're doing, the cells are opening, and the foreign pathogens or bacteria or viruses, they are engulfing it, they are eating it, they are getting inside. Now in this stage 1 you can see it start to engulf, and in stage 2 it completely engulf and the mouth has been closed, and in phase 3 here the bacteria or virus completely get inside the macrophage. So in this fashion, from this point to this point, you can see that pathogen has been completely eat by this macrophage, completely engulfed by this macrophage. It is a immunity bound reaction. It is just a response to the immunity. If our immunity will be activated at that time, the engulfment by the macrophage cell will occurs. In the another say in another slide, or in the another figure, the pinocytosis will occur. What is pinocytosis? It is a process of drinking. Here eating, here is the drinking. So drinking of what? Drinking of the droplets of solutes, droplets of viral solutes which are required for their process, or which are required for their further requirement, for their further movement, or for their daily requirement, whatever things are they want for their regular processes. So these droplets from the outside, they are get entered into the these vesicles, and the sac will be filled, and here closing will occurs. These large molecules are engulfed by this cell, and after that they can get entered, and here in the vesicle you can see the droplets of that solids are available. It will be nutrient, it will be salt, it will be electrolyte. Now it can be taken or utilized by the cell for their further function. So in this fashion the pinocytosis will occur. There is too much difference between pinocytosis and phagocytosis. Both are different. Here we are taking the non-living things, and here you are taking the living things. They are the bacteria, viruses. They are the living things. They are engulfed. This occurs for the immunity purpose, immunity boosting mechanism, and this is the regular, uh, regular performing mechanism for their cell growth, the cell development, or cell regular process. It occurs here. So in this fashion, we have discussed this process of endocytosis. Now, forms of endocytosis, as we have discussed, phagocytosis is nothing but the cell eating, and the pinocytosis is nothing but the cell drinking process. The droplets of these electrolytes or nutrients are drink. Now, exocytosis. So, what is exocytosis? It is nothing but the process of throwing outside or movement of the internal part of your cell to go outside, to throw outside. So that process is called as exocytosis. Reverse of endocytosis. It is just opposite process of endocytosis. and the cell discharges the material. Means due to the budding or breaking of that cell lines, the whatever the unwanted materials are there inside our cell into the our secretory vesicles, so they are throwing it outside. The same process also occurs in case of the neurotransmitters. When neurotransmitters are released from at the synaptic vesicle through neuron, so in the same this secretory vesicle, the here the neurotransmitters are available like acetylcholine or noradrenaline or ACH or dopamine, and they are collected in the sac. And here, after the opening the close of that cell, the neurotransmitters will go outside. The various chemicals are available here, so they go outside and they will go to the respective place. So in this fashion, the process of exocytosis will occur. So cell discharge some material, so material in the form of chemical, in the form of neurotransmitters, in the form of waste materials also. Waste materials also throw outside through the cell by the process exocytosis. So I hope you understood the process of exocytosis. There is difference between exo and endocytosis. They are oppositely different processes. Now here, the vesicles moves to the cell surface and the membrane of vesicles fuses. The members of vesicle, these vesicles are coming towards the surface from 1 to the 2, and they are fuses to the surface. And at the at the surface the budding will occur, and these cell lines or the border of the cell will start to vanish, start to break, and after that whatever the internal these materials are there, they are expelled outside. So in this figure you can see they are expelled outside. So students, in this fashion, we have discussed the transportation of the solutes across the cell membrane. So how it occurs in our body, what are the various processes occurs like diffusion or osmosis or endocytosis, exocytosis, and how this transportation occurs in our heart, in our kidney, how they are constantly doing in our body. So that we have discussed. If you have any queries, you can ask me, you can discuss with me. So till then, have a nice day. If you have any queries regarding this, so you can ask me. In the next lecture, we will discuss about the cell division.